Welcome to the Diary of a Medicinal Chemist.
We have discussed about the drug discovery stage and, of course, a number of activities that are carried out in the drug discovery stage. Now, today we'll be looking at the drug development stage. And the first step or the first activity that is carried out in the drug development stage is the preclinicals. Preclinicals. Preclinical trial. And what does this mean? This is simply trying out our potential drug, our drug candidates on animals, carrying out animal studies. We have a number of different tests that are carried out in order to assess the pharmacokinetics and pharmacodynamics of the drug. Because a number of animals share a high level of semblance in terms of their metabolic machinery to humans, and that is why most times we believe that most of these the results that are gotten from these animal models can, of course, give us insights into how the drug would also perform in the human system. And that is why essentially a number of tests are carried out on rats, on rabbits, on a number of other animals to ascertain the pharmacokinetic studies and the pharmacodynamic studies of the drug candidates. Now, the preclinicals, we are just as I mentioned, we are making use of animal models, trying to look at the toxicity level, trying to look at the distribution of the drug, trying to look at the absorption of the drug, and of course, trying to look at the clearance rate of the drug, and a number of other endpoints which are very key in drug discovery. Then afterwards, we move to the clinical trials. Move to the clinical trial. The clinical trials are in four phases. We have the phase one, and what we do in phase one is essentially looking at the human pharmacology. Human pharmacology. Essentially looking at this is the first time we are testing the drug on human subjects, and the size, the population that are administered, that the drug is of course administered to are very small. You can have from 10 to 15 volunteers with their consent, of course. Now, at this level, we are trying to look at the pharmacokinetics and pharmacodynamic studies. We are trying to look at the tolerance level in the human system. We are trying to look at the toxicity and so many other endpoints, trying to get a feel of how the drug will perform. And again, it is essential to note that the volunteers that are used in the phase one are normal patients, patients without that particular disease that the drug is designed for. So they are normal patients. We just want to have an idea of the pharmacokinetics, what the body does to the drug in terms of from the absorption to the distribution to the metabolism to the excretion. In the excretion, we are looking at the glomerular filtration rate, we are looking at the creatine clearance rate, volume of distribution, and so many other very important parameters that we have discussed about in the ADME studies. Now, after the human pharmacology, which is the phase one, we have the phase two, and basically what we do in the phase two is basically therapeutic, exploratory. So phase two is basically hinged on therapeutic exploratory, trying to look at a number of other things in terms of dosage, in terms of the dose response curve, and so many other endpoints that were not catered for. And again, the volunteers in this stage are way are more in the sense than the phase one. And again, these are volunteers that have given their consent to be available as subjects for the testing of the drug. And